Welcome to Angel's online platform 12th mega test, date 29 September 2025. Question number 91. One of the possible early source of energy were option CO2, chlorophyll, green plant and UV rays. Correct answer is UV rays and lightning. Question number 92. According to Operin, which of the following were not present in primitive atmosphere of the is? The correct option is B. Oxygen was not present in early or primitive atmosphere of Earth. During the purification process for recombinant DNA technology, addition of ice-chilled ethanol to precipitate out DNA. So option 1 is correct. In the solar system of Milky Way galaxy, Earth was supposed to have been formed about. So Earth was formed about 4.5 billion years ago. So again answer A is correct. Evolution is progressive development of race, history and development of race along with variation, history of race, and is a random change in gene frequency. So evolution is a history and a development of race along with variations. Question number 96. Who demonstrate that life come from life with help of flask with boiled broth? So Louis Pasteur conducted a snake flask experiment. Operin theory is based on, so it is artificial synthesis experiment to prove that synthesis of organic compound formed the basis of origin of life was performed by. So this experiment was carried by S.L. Miller. So A is right answer. Which of the following amino acid was not formed in synthesized in Miller's experiment? Okay, so glycine, alanine and aspartic acid were formed while glutamic acid were not formed. So option A is correct. Miller performed experiment to prove a biogenetic molecule evolution of life. Which molecule was not present in Miller's experiment? So water, methane and ammonia was present, but oxygen was not present in Miller's experiment. Okay, match the column. So Darwin, origin of species, so A2, okay, दो में है. Then Lamarck, so use and disuse of organ, B1. So again option A is correct. Okay, C, Hugo de Vries, and he gave mutation theory, and Operin gave concept of origin of life. Okay, Miller synthesized a simple amino acid from. So Miller synthesized simple amino acid from hydrogen, methane, ammonia and water. So B is correct. Life cannot originate from inorganic material at present or currently because very high amount of oxygen present in a atmosphere. Question number 104. According to Darwin, fitness referred to, so according to Darwin, fitness referred to reproduction ability and ability to survive. So A is correct. Match the column, find out the correct combination. Okay, so again Charles Darwin, origin of species, so A4. Lamarck, acquired characters, so B3. Then Hugo de Vries, give mutation theory C1. Then Ernest Haeckel give biogenesis genetic law, and Weissman give germplasm theory. So E2. Okay, so correct option is D. Based on the observation, Charles Darwin concluded that existing life forms have similarity with the life form that exist million years ago. Evolution history, he suggests. Which of the following is our condition, our connotation of the theory of special creation? So all living organisms were created as such. Diversity will remain same in the future, and Earth is about 4,000 year old. So all of this correct. Darwin explained origin of species through natural selection and branching descent. Darwin theory of natural selection was based on, so it is based on enormous rate of reproduction in organisms, struggle for existence, and survival of fittest. The finches of Galapagos Island provide an evidence in favor of, biogeographical evolution. They provide evidence. Which one is important consequence of geographical isolation? So species formation through reproductive isolation. Example of convergent evolution are, so sweet potato and potato are example of convergent evolution. Other are example of divergent evolution. Which one of the following is most important for speciation? So reproduction isolation is the main point for speciation. Which of the following site has homologous organ? So mouth part of the cockroach, mosquito, and honeybee are example of homologous organ. In some organism, the existence of analogous structure, wings of insect, bat, joint leg of insect, and a vertebrate, is due to phenomena of, so it is a phenomenon of convergent evolution. They are convert from one another. Fossilization can occur where animals are buried and preserved by natural process. So A is correct. Industrial melanism is example of, it is a protective reassemblance of surrounding. Analogous organ are wings of bird and bat and butterfly. They are analogous organ because they all are used in flying. When two species of different geology come to reassemble each other as a result of adaptation, the phenomenon is termed as, so it is termed as convergent evolution. Evolutionary convergence is characterized by development of common set of characters in a group of different ancestry. So option B is correct. The diversity in the type of beak of finches adapted to different feeding habit on Galapagos Island as observed by Darwin provided evidence for origin of species by natural selection. Adaptive radiation refer to evolution of different species from common ancestor. B is correct. During the process of gene amplification using PCR, if very high temperature is not maintained in beginning, then which of the following step of the PCR will be affected first? So, if in a beginning high temperature is not maintained, the denaturation is not occurred. Question number 124. Darwin finches from various islands of Galapagos Island different in a size and shape of beak, यहाँ पे beak आएगा, is due to it is due to adaptive radiation again. Which of the following statement would Darwin agree with? So individual width and population vary in characteristic they possess. Evolution is the best view as purposeful and directed change over time. Natural selection is mechanism by which biological evolution take place. So here D option is correct. All of the statements are correct according to Darwin. Tasmanian wolf and is marsupials, while wolf is placental mammal. This shows convergent evolution and adaptive radiation. The correct order of steps in a PCR. So denaturation, annealing, and extinction are correct order. D option is correct. The factor involved in formation of new species are isolation and variation. Lamarck theory of evolution is also called. So it is also called inheritance of acquired characters. Mutation according to a mutation discovered by Hugo de Vries are. So mutation are random and directionless. Transfer of gene from one gene pool to another is called gene flow. Hugo de Vries gave his mutation theory of evolution while working on organism organic नहीं यहाँ पे आएगा organism. So organism is Oinothora lamarckiana. Which extinct animal shows evidence of evolution? So Archaeopteryx is correct answer. De Vries theory of mutation is opposed to natural selection, not opposed to natural selection, opposed to germplasm, and opposed to Lamarck theory. So it is not opposed to natural selection theory. Stabilizing selection favors intermediate form of trait. Genetic drift operated only in So it is operated in a small population only, or we can study in small population only. Heidelberg equation is affected by gene migration, gene flow, genetic drift. So all of the above. Okay. Again match the column. Wallace. So Wallace is a co-proposer of natural selection. Malthus write essay on population. So B is 1. Heidelberg gave equation. So C3, and industrial melanism is Bitson betularia is a moth. So again option D is correct. DDT resistance in mosquito is an example of. So it is example of directional selection. Random genetic drift in a population probably result from so interbreeding with a small population. Question number 141. You have to identify the homology in a diagram. Okay, so here a thorn of bougainvillea, and this is a spring like a structure of cucurbita. Okay, so P and R is a correct option. Tendrils. In the given figure is example of so it is example of both homologous organ and divergent evolution. Identify given diagram. So first one is stabilizing selection, second one is directional, and third one is disruptive. Okay, so option B is correct. The following diagram represent question number 144. The following diagram represent Darwin finches represent adaptive radiation and convergent evolution. So option D is correct. Again diagram represent homologous organ. Who gave definition of biotechnology? So European Federation of Biotechnology gave definition of biotechnology. Dash technique has given in birth of biotechnology. So genetic engineering and maintenance of sterile ambience give birth to biotechnology. Which of the following enzyme is known as molecular seizure? So restriction enzymes are molecular seizure. When does first recombinant DNA was developed? So it is developed in 1972. Which structure is involved in genetic engineering? So plasmid is used in genetic engineering. Genetic engineering aimed at destroying wild gene, replacing defective gene, curing human disease by introducing a new gene. So all of the above. The procedure through which piece of the DNA is introduced in a host bacterium is known as transformation. EFB stands for European Federation of Biotechnology. Plastid used in construct first recombinant DNA was isolated from which bacterium species? So first DNA recombinant DNA was constructed from Salmonella typhi murium. Extra chromosomal self-replicating double-stranded circular DNA in bacterial cell is called plasmid. Select the wrong statement. Developing DNA vaccine is example of biotechnology, correct. Artificial recombinant in 1972, correct. Replication of DNA initiate at promoter site, so it is wrong. It is from origin of replication site. And replication of plasmid independent of control chromosomal DNA in bacteria, so it is correct again. Read the following statement. Biotechnology is integral of natural science and organism cells, part thereof and molecular analog for production and service. So correct statement है ये. DNA can be chemically altered by genetic engineering. Again correct. Recombinant DNA use DNA polymerase of its host for replication. Correct. Cohen and Boyer reassemble for first artificial synthesis of RNA. So here RNA is wrong. It is DNA. So it is wrong. Replication of the DNA initiate at origin of replication. So again correct. The four statements are correct. Sexual reproduction result in introduction of undesirable variations as well as desirable variation. So option D is correct. Genetic engineering has been made possible due to observation of DNA under electron microscope. We can break DNA at specific point. Ability of restriction endonuclease in purified form and knowledge of transduction. So again C option is correct. Which of the following result in growth of desired organism in biotechnology? So genetic engineering, maintenance sterile ambience and use of restriction enzyme. Which of the following is result in growth of desired organism? So D option is correct. All of the above technique is used. Which technique result in introduction of one or more desirable gene into specific host? So through genetic engineering we can do this. Which enzyme joins sticky end of DNA? So DNA ligase is correct answer. Specific region or a sequence of the DNA which is responsible for initiating replication. So origin of replication is correct answer. Which vector can clone only a small fragment of DNA? Answer is plasmid. Biolistic technique is used in gene transfer process. Which type of bonding facilitate action of DNA ligase? So with help of hydrogen bonding. Which of the following help in visualizing DNA fragment? So in agarosal electrophoresis we have to use TBR and exposed to UV light. So correct answer is D, both B and C. Question number 168 dash. Protein involved in replication of the protein. The replication of protein is through rope gene. A restriction endonuclease break bond between sugar and phosphate component of nucleic acid molecule. gene rope present in PBR322 codes for protein involved in translation, protein involved in replication of the plasmid is correct option. In PBR322, tetracycline resistance gene has recognition site for which of the following enzyme? So in tetracycline BAM-H1 site is present. Which of the following tool is not a tool for genetic engineering? So GMO is not a tool, it is a result of genetic engineering. All the following are basics tool for recombinant DNA technology except the cloning vector component host restriction enzymes are tool, but RNA polymerase enzyme is not required. Palindromic DNA sequence of EcoR1 is, so it is a G 5' to 3' GAATTC, so C option is correct. PBR322 has two antibiotic resistance genes. These are against ampicillin and tetracycline. Genetic engineering would not be possible if one of these were absent, so answer is DNA ligase. Restriction enzyme cut palindromic DNA sequence at little away from the center and between the same base on opposite strain, so C option is correct. In PBR322 EcoR1 site is located in between ना EcoR1 ऊपर है तो in between tetracycline and ampicillin resistance gene. Identify ideally in process of genetically modified organisms a vector should have recognition site, so we can do a cloning. Last question, A can transform normal animal cell into cancerous cell, so here retrovirus is answer, and while B can transform normal plant cell into tumor, so TI plasmid can do this, so answer is B. Hope you do well in your exam. See you tomorrow. Bye.